My name is Natalie Galusha, and I am the manager of the Center for Aging. With me here today is also Nancy Morrow-Howell, our director. You will see here at the end, she'll be announcing our winners. So as we get started here for our 3MT competition, I'm just going to give a little bit of background about the competition, explaining a little bit about how it works, and then we will go ahead and kick things off. And thank you also, would like to recognize the people who have joined us on Zoom today. It's been nice to be able to have this hybrid option so people who aren't here locally or just weren't able to make it this afternoon could tune in as well. And we will have an option at the end, which I'll be explaining more later, for people's choice voting, and we'll be doing that on Poll Everywhere, which I'll have that on the slides, but then I also put the login information up here for that as well, in case you're familiar and want to go ahead and get that set up. So just to get us started here, a quick thank you to our sponsors, of course, the Friedman Center for Aging, and then also the Foundation for Barnes-Jewish Hospital, the Harvey A. and Doris May Hacker Friedman Fund, which helps us to be able to provide the monetary awards for this competition. All right, so just a little bit about what this competition is, if you aren't familiar with it. So this competition is called a 3-minute thesis, which is a research communication competition that was developed first by the University of Queensland. So all of our PhD students today are going to have just 3 minutes to present their thesis to you. Obviously, as I'm sure many are familiar with, theses are much longer than that. And so really it is up to them to be able to portray their message so that everybody can understand it here in the room. It is an international competition, so this is just a quick little snapshot of where some of these competitions have been held. This is the third time the Friedman Center for Aging is doing it. The Lieberman Graduate Center at WashU also does it, and I'm sure there are other universities in the St. Louis area and other areas at WashU that do it as well. And we are also joined today by our judging panel, which is in the very front here. We have Dr. David Belota, who is in psychological and brain sciences, Madeline Franklin, who is the executive director of STL Village, a local organization here to St. Louis that serves older adults and assisting them with aging in place. And then Carolyn Baum, who is a professor of occupational therapy and is also in neurology and social work. So thank you to our judges for being here today as well. So a little bit more about the rules of the competition. Each presenter today will have a single static PowerPoint slide. There's no transitions or animations or anything like that. No electronic media, no props. And as I've mentioned already, each of the presentations are limited to a maximum of just 3 minutes. We're not going to cut them off in the middle of the sentence, as I've explained to them, but are going to try to hold them as accountable to that 3 minutes as we can. And all of them are required to be in spoken word. Will be presented up here, either at the podium or using a handheld mic. And they will begin as soon as they have transitioned to their slide. And then the judge's choice winner will be chosen by our judges panel. So just a little bit of a quick overview of the judging criteria so everybody knows what the judges will be looking at. They will be looking first at comprehension and content. So they'll be looking at things such as, did the presentation clearly describe the impact of the research results? Did it follow a clear and logical sequence? And did they spend an adequate amount of time on each element? The second category that they'll then be looking at is engagement and communication. Did it make the audience want to know more? We will have a little bit of a brief Q&A. Was the presenter careful not to trivialize or generalize their research too much? Or did the PowerPoint slide enhance the presentation? Was it clear, eligible, and concise? So a bit on that as well. And these are going to be our prizes that will be awarded at the end. So we will have the Judge's Choice winner will receive a $1,000 cash award, and the People's Choice will receive a $500 cash award. And the awardees will be recognized and present their 3-minute thesis again at our 21st Annual Friedman Lecture and Awards on April 13th here on the medical school campus at the Eric P. Newman Education Center. All right, so just a little bit of our agenda and how things are going to work today before we get started. So we're going to do part one of our presentations. We're going to have 4 presenters present. So they'll each have their 3-minute presentation, and then we'll have a couple of minutes for about 1 to 2 or 2 to 3 questions from the audience, as well as those on Zoom. So those of you who are participating on Zoom, please be sure to put your question in chat, and we will have somebody here in the room ask that question aloud to the presenter for you. And then after we have a couple of minutes for questions, then we will proceed on to the next presenter. After the first 4 presentations, we'll then take a short break and then have our second set of presentations. And then that's when I will come back up here, explain a little bit more about how the People's Choice voting works. We'll take a break again to allow the judges time to deliberate and choose the winner, because it is based on the scoring, but then there is a bit of discussion that happens because sometimes there's a tie or other things that happen as well. So we give them a bit of time to discuss. So we'll have some time here in the room for a bit of networking while we're doing that. And then at the end, we will announce our winners and wrap up. All right, so with that, we are going to go ahead and kick things off. And first we have Jordan Kassab, who is an applied health behavior research program, and she is going to be presenting frailty as a potential early sign of Alzheimer's disease.
What? Do I change the sound?
Great.
I got it. Are you ready? Yes. Okay. I'd like to start off by posing a question. How can we make treatments for Alzheimer's disease more effective? We could pour millions of dollars and resources into finding new treatments and new drugs, or we could find a way to start these interventions sooner. Now, that's certainly easier said than done. Currently, more than 6 million Americans suffer from Alzheimer's disease, which is a degenerative mental disease in which elderly adults have trouble with memory and an inability to maintain their current lifestyle. Preclinical Alzheimer's disease is a cognitively asymptomatic stage that precedes Alzheimer's disease. In order to be diagnosed with preclinical Alzheimer's disease, physicians look for protein deposits as biomarkers in the brain indicating physical changes. Once these biomarkers are already present in the brain, dementia and cognitive decline are likely to ensue soon after. Past research has shown a very strong link between physical and cognitive health, and this exact relationship is what inspired my specific study. Can physical precursors be a potential sign of Alzheimer's disease? If we can detect someone, if they are on the path to developing Alzheimer's disease, maybe we can start interventions sooner and delay the onset of Alzheimer's and maximize the good life for elderly adults. In this study, we enrolled hundreds of elderly adults with normal cognitive functions. The patient cohort included both those with and without biomarkers indicating preclinical Alzheimer's disease. However, none of the participants were made aware of their biomarker status. We used a myriad of physical and lifestyle assessments in order to identify any potential risk factors for these elderly adults. And one of these tests is the grip strength test. So a grip strength test is a common test used to assess overall bodily strength. And it's used by using a hand dynamometer. The participant squeezes as hard as they can, and a numerical value of how much weight they can grip is given. In this study, we have shown that while controlling for age and gender, that those with preclinical Alzheimer's disease have statistically significant lower grip strength scores. So although no dementia has onset, physiological changes have already begun. So the overall aim of this study is to identify any potential factors that take precedence before the dementia starts. The clinical implications are clear and strong. More research needs to be investigated in order to identify any potential candidates of preclinical Alzheimer's disease sooner. By doing this, we can start interventions early, and we can delay and potentially even stop the devastating fate of this neurodegenerative disease. Thank you.
All right, does anybody have any questions for Jordan? Nancy?
Yeah.
Hold on, wait till Michelle gets you that. Yeah.
There we go. Thank you. And to clarify for the audience, this Q&A is to give the judges a little breathing room. It's not part of the judging per se. Jordan, what is why the mechanism, the pathway between physical and cognitive here? And what's your hypothesis about which comes first and why they're related?
So my hypothesis is that physical precursors occur before cognitive precursors. This has been shown in other diseases, and so it's not been a complete, the pathway has not been completely proven in Alzheimer's disease yet. So that's hopefully what I'm trying to work towards, is showing that there are some potential precursors before that cognitive decline, so we have to see that.
Mhm. But is your idea that the causes of the physical decline or weakness is stemming from the cognitive disease?
Potentially with the biomarkers and the protein deposits could cause potential muscle deterioration or so, but I still believe that it will probably start before the dementia.
Yes.
Hi, so I'm just curious to hear more about the other measures going on here, because like we know from past work, like grip strength is again a correlate, but not a very strong correlate of cognitive functioning. So in terms of intervention, that doesn't that gives us a minor idea of what we might want to do. That grip strength isn't such a great predictor, but are there like from the broader bevy of inventories you have here, what kinds of things seem to be stronger or weaker physical functioning predictors?
Yeah, so some of the other physical tests we worked on were some balance tests using a V-Trax device. And that was also shown to be a correlate with this cognitive function. Additionally, we have, like I was saying, the lifestyle test, they have a PHQ analysis for a depression scale, and those are just more behavioral or so, but a lot of the tests are balance and strength.
Thank you. All right. From Psychological and Brain Sciences, who is going to present on sleep quality and memory across the lifespan. Have you ever struggled with a problem that you just couldn't solve, and someone tells you, you should sleep on that? Well, it turns out this might be good advice for several reasons. Sleep reduces our stress, it improves our focus, but also sleep helps us piece together information into memories. Unfortunately, as we get older, both our sleep quality and our long-term memory abilities get worse. Older adults experience more time awake during a night of sleep and experience less deep restorative sleep. And there's evidence that these changes in sleep actually may lead to changes in memory in later life. However, many of these studies have focused on simpler forms of memory, such as the ability to remember a pair of words. But what about more complex memory abilities, such as our ability to remember a spatial layout of an environment? Each day, we use this ability to find the location of objects or places. For example, you might need to remember where the train station is or where your car is parked after leaving this talk. This type of memory is selectively impaired in older age. Older adults who report more difficulty finding their way are also more reluctant to travel to new locations. And this decrease in mobility can lead to reduced independence, reduced quality of life, and reduced subjective well-being. That is why understanding the factors that may cause this decline, especially factors that we might be able to change, like our sleep, is so important for preserving our way of life in later years. The goal of my research is to understand the relationship between age, sleep, and memory of a spatial environment. In an earlier study, I measured sleep using a wrist activity monitor, similar to a Fitbit or an Apple Watch, in both young and older adults. And then I measured their memory. I found that overall, older adults had more difficulty remembering the spatial layout of an environment. But older adults who experienced more awakenings in the night experienced even greater difficulty remembering this environment. One potential explanation for this is that older adults who wake more frequently
experience less deep restorative sleep. My future research is aimed to measure this directly by using a device that detects electrical signals from the brain during sleep. From this, I can determine whether changes in brain activity during deep sleep predict older adults' ability to learn and remember a spatial environment. By gaining this knowledge, we are one step closer to understanding and preventing the decline in spatial memory that occurs in later years. Hans Christian Andersen said, "To travel is to live. Perhaps a key to living a longer life lies in a good night of sleep."
Just gonna keep that, yep. All right, any questions for Hannah? Emily, anything on Zoom yet? Okay. Pat?
Hey, Anna, great talk. So I know this is about the award on aging, but looking at the chart, I guess, could you speak to the young adults a bit? Like, it seems like they're relatively intact, and that's kind of a surprising finding, I guess, in some respects.
So they were intact in terms of their ability to remember the layout of an environment, which is about what we would expect. This is, these are adults, undergraduates who are, 18 to 23 years old. So they have a pretty good hippocampus. But yeah, so the interesting finding is that we don't see this relationship between the sleep and the spatial memory in the older group that we do with the younger group. So it could reflect an underlying vulnerability to sleep that you see in age. Perhaps there's other factors that provide resilience in younger adults.
Sure, yeah.
Thanks, Hannah. That was terrific. How do you, there's lots of reasons why we sleep worse in later life,
Yeah.
right? So how do you control for all of these other confounds that might be involved in this relationship?
Yeah, that's a great question. I think this is especially one of the motivations for using the device to measure brain activity directly. Because in sleep, perhaps there's reasons for disruptions. You might have more pain. There's changes in urinary function, all of those things that could lead to more disrupted sleep. So I think that the direct answer would be to be able to look at brain activity and use that as a potential correlate for, that this is an underlying mechanism related to cognition.
Thank you, Hannah. All right, and next up is Lucia Capano in the Division of Biology and Medical Sciences, and she will be presenting why capturing age matters, advancing our understanding of neurodegeneration.
Let's imagine when you're born, you're given a brand new fluffy teddy bear. It's yours and it's the only one you get for your entire life, and you have to keep it. When you're young, it's just you and Teddy hanging out, doing things. And as you old, well, Teddy starts to show some wear and tear. Now, these bumps and bruises can lead to an increased risk of a catastrophic event. And there I'm talking about neurodegenerative diseases, specifically tauopathies, the most common form of which is Alzheimer's disease. Tauopathies affect over 30 million people worldwide. And despite 30 years of research, we still have no treatments or preventions for this disease, and it is always fatal. How can this possibly be? In my opinion, it's because scientists have been using the wrong teddy bear to study the disease. Let me explain. In our analogy, if we want to know what happens to cause a hole in our beloved teddy, we would want to study an old, worn-out teddy bear. Now, in the real world, this would be getting our hands on adult, alive human brains. And I bet you can imagine that's hard to come by. So scientists came up with a way of making brains in a dish, but they make them from stem cells. And here's the crux. Those cells that they make are now young, the same age as a baby. And so if we want to know what those weaknesses do to cause a potential tear, a brand new teddy bear is not going to get the job done. Here's where my thesis comes in. I make old brain cells, AKA those bumped and bruised teddy bears, and they perfectly mirror what we see in the adult brain. I do this by taking human skin cells and giving them 2 small guides called microRNAs. These instructions tell the cell to stop making those skin cell genes and to start making the brain cell genes, giving me a perfect, well-worn teddy bear. So when I made cells from patients who have tauopathies, I was able to see the clumping of proteins that are hallmarks of the disease. That's never before been seen in a human cell model. And so with this system, I hope to be able to parse apart what's going on in that aged, worn out teddy bear so that no one has to suffer from tauopathies again. Thank you.
All right, questions for Lucy? I have a simple one. How did you find such a perfect picture to go with your?
An excessive amount of Googling. Yeah. Way too many hours.
Yeah. Perfect, thank you.
Yeah, so a question about just your experimental setup here. So I'm curious if you have like any control cell types that you also treat with microRNAs that you would not expect to get tauopathies, and if you control for that.
Yeah, great question. Okay, so yes, whenever I do my experiments, I have cells that come from someone who doesn't have a tauopathy and someone who does have a tauopathy. What I see is those who don't have tauopathies don't show me that clumping. Those that do have tauopathies show me that clumping. So I always have that negative control there. Great question. So it's not the microRNAs, that tool that's giving the clumping. It's something that's built into the cells themselves. Colin, or sorry, Jason.
Thanks. So what kind of questions are you trying to ask with your cell model? Are you trying to see what causes the clumping or what can help to unclump the tau protein? So basically, what kind of questions are you trying to ask?
Yes. Yeah, absolutely both, right? We want to know why are we getting these clumps? We want to know how we can get rid of these clumps. Yeah, totally. Both.
One more question? Anybody? All right, thank you, Lucy.
Thank you.
So next up, we have Jared Elenbaas. He is with the School of Medicine and Internal Medicine. And he is going to be presenting exploring how a protein biomarker of aging contributes to age-related human disease.
Okay, thanks for that introduction. Okay.
Go ahead.
All right. Thanks everybody for the opportunity to present my thesis work. So I'm actually taking a little bit of a different approach here than some of the other contestants. And I wanted to talk to you about how we're exploring how a protein biomarker of aging actually contributes to age-related disease. So we got interested in this protein called SFEP1 because it increases during aging. And it doesn't only just increase in aging, it actually accelerates during aging. And it accelerates faster than any other protein in the plasma, which is really quite striking. This protein is also associated with heart disease, with high blood pressure, diabetes, with dementia, glaucoma, and longevity. So it has a ton of really relevant disease associations, especially for elderly patients. But as this audience knows, correlation doesn't necessarily mean causation. And so we used this sophisticated genetic tool where we looked at genomic data and proteomic data and asked whether this protein actually causes disease. And you can see by the positive slope of this blue line here that in fact, this protein does cause disease. So we use similar techniques to look at receptors for this protein within plasma. And we identified a cell surface protein called PAIR1. We asked the same questions about pair 1, does it actually cause cardiovascular disease? And you can see here in the green line that indeed it does. So then when we look at cells and combine these two proteins together, we notice that it results in the activation of aging-related pathways, such as the pathway called mTOR. And we think this might be the mechanism by which both of these proteins actually cause a number of these age-related diseases. And so our hope with this project is that we'll be able to characterize this interaction and potentially develop drugs to block this interaction, hopefully resulting in decreases in a number of these different diseases, and also ultimately promote healthy aging. And so we're really excited about this project, and the university is excited about it too, and it's actually filed a number of patents, which is really exciting. And so I'm really happy to be able to share it with you, and I'm happy to take any questions as well. Thanks.
Thank you. Yeah, so where do you have any idea about where the SVEP1 originates and how it gets into the blood? Like which kind of cell types are involved with that?
Yeah, so that's a really great question, and it's actually pretty difficult to answer. So you may be familiar with this concept of like taking a liquid biopsy. And the thinking there is that a lot of proteins simply leak from the tissue that they originate in into the plasma. And so really we're thinking about tissues that produce a lot of this protein. And those tissues include, for example, adipose, so fat, as well as vascular tissue. And really a lot of the disease associations that I was discussing here are strongly related to vascular health. So we suspect a lot of it's coming from the vasculature.
Okay, Emily, yes.
Sorry, we have a question here from one of our virtual audience members. Do you believe that diet and lifestyle affect these proteins, helping to deaccelerate the generation of this protein?
Yes, so diet and lifestyle almost certainly affect the amount of this protein. And one of the strongest predictors for the plasma levels of this protein is BMI, or the amount of weight that a patient has. And so that is definitely a factor that contributes to the amount of protein that is in plasma, yes.
One more quick question. All right, thank you, Jared.
Thank you.
All right, that brings us to Ariella Coler-Reilly in molecular cell biology. And she is presenting is the answer to longevity hidden in our genes. Oh, yeah. Okay. We're just getting the red guy out of the way.
Yeah.
Yeah, testing. I'm Ariella. Can everyone on Zoom hear me? Okay. So we all know that as we get older, we're destined to experience a decline in health. Our hearts may fail, our bones may weaken, our minds may even deteriorate. According to US Medicare data, 80% of adults age 65 and older are diagnosed with at least one chronic disease, and often more than one. The goal of my research is to use genetics to try to figure out why. So our gene sequences don't really change with age. If you get your gene sequence when you're 5 years old, and then again when you're 90, the results are going to match. Nothing's really different. However, what does change is how your body uses those genes. There's a term called gene expression, and that indicates how much a gene is being used. So as you can see illustrated at the top of my slide, scientists in my field for years have been taking samples from young and old individuals and comparing the gene expressions in various different organs, trying to find differences, and in the hopes that we can use these differences to understand the mechanisms of aging and prevent or even reverse some of this decline. The problem is that even when we identify these genes, we don't really know what to do with them. Think about it. If we find a gene and the expression of that gene or how much it's being used has increased with age, maybe that means, yes, this is what we're looking for. This gene has gone rogue and is causing aging. Sorry, it's a driver of aging. But it could also mean the exact opposite. Maybe that gene is actually more active because it's responding to stress and trying to protect us from aging. So it could mean two completely opposite things. So for my research, my thesis, my goal is to design a filter so that we can figure out which genes are actually playing a causal role in aging, and if we have the power to change that. So the way that I did this is I looked at data from 25 of those studies that I mentioned before, and I calculated which genes were flagged most often across all of those studies. And then I designed two different experiments that are complementary approaches to figure out which ones had a causal relationship. So in worms, I wanted to look at lifespan directly, and this is very easy to do in worms. And so I changed the gene, I changed a gene in a batch of worms. I changed each gene in a different batch of worms and measured how well those worms thrived and if they lived longer. And then I also wanted to directly test the genes in humans, but it's illegal to do genetic tests on live humans. So instead, I harvested bone marrow and isolated stem cells from that bone marrow and tested the genes on those. So ultimately, in the end, I should end up with genes like this purple gene in my diagram that have been proven to keep worms and stem cells living happier and healthier. And I hope that eventually we can also live happier and healthier. Thank you.
All right. Any questions? Nancy, you want to go first?
Thank you so much, Ariella. This is way outside what I know about. Can you give me an example of a certain gene, a gene expression that leads to a certain disease?
Sure.
A changing gene expression that leads to a certain disease in later life.
So that's a very, very good question. There are lots of genes that are associated with different diseases. I think one of the most famous age-related genes is maybe ApoE. A lot of times there are mutations in certain genes and that affects their gene expression. So gene expression is really correlated, is really strongly related to mutations. So if you think of any disease that's caused by a gene mutation, that's also related to gene expression, because it could be a type of mutation that sort of messes up the gene called loss of function. So that reduces the gene expression, or a gain of function, which means that it's going to increase the expression. So yeah, pretty much anything that has a genetic component, including Alzheimer's.
Okay, that really helps, because I also get stuck on how does the expression change? Is it always in a certain direction, in a certain way?
It really depends on the gene and the organ. That's why it was really important in my research. As you can see in the cartoon, the samples are being taken from lots of different organs. And so it's really important to look across the whole organism so that you're not just getting a gene that's very specific to, say, heart function. So the expression may go up, it may go down. It depends totally on the gene, the organism, and the tissue.
Simple.
Yeah.
Sorry.
Do you have one, Emily?
Yes, I believe we have a question. So is the point to identify gene expressions that lead to longevity in general, or gene expressions that lead to diseases that reduce longevity?
Those are two very related issues. I think a lot of people make a false dichotomy between longevity and diseases that are associated with age. And the reason that you die is normally because you got a chronic disease of aging, like heart failure or dementia, or you fell because your bones were weak and then had to do a surgery and then you died in surgery. So there's kind of a false dichotomy between what we call health span and lifespan. Ordinarily, any intervention that extends lifespan, we've never found one where it makes you less healthy. They always go hand in hand, at least so far.
Thank you. All right. Thank you. Okay, and now we have Gabrielle Pfund, who is also from Psychological and Brain Sciences, and she's going to present on the roles of time and measurement and within-person purpose variability.
Hello. Okay. When you consider your next hour, day, week, or month, and you ask yourself, are the things I'm doing worthwhile? Are the activities I'm engaged with meaningful to me? How I'm spending my days, do they match with my true interests and values? Depending on how you answer that question, this gives us an idea of your sense of purpose. Sense of purpose can be understood as the extent to which one feels that they have personally meaningful goals and directions guiding them through life. And we care about this construct because it's tied to a wide range of desirable outcomes throughout the lifespan, and particularly in older adulthood. When it comes to well-being, people with a higher sense of purpose experience more positive emotions, fewer negative emotions, are less reactive to stressors, and are generally more satisfied with their life. When it comes to physical health, people with a higher sense of purpose are at lower risk for physical disability in older adulthood, heart disease, and stroke. Cognitively, for older adults, people with a higher sense of purpose experience higher cognitive functioning, slower cognitive decline, are at lower risk for Alzheimer's disease and mild cognitive impairment, and people with a higher sense of purpose live longer. So we know that sense of purpose is associated with a wide range of desirable outcomes, many of which have been discussed today. But an issue that we run into with this construct is that it often declines in older adulthood. And this happens following things like retirement, cognitive and physical health declines, as well as losing loved ones, like spouses and friends. So this puts us in a position where we want to help older adults maintain a sense of purpose, but there's a gap in the literature regarding how we can do that. That's where my dissertation comes in. The big gap in the literature right now regards the fluctuation of this construct within an individual. So at the beginning, I asked you how you would respond to those questions at the hourly, daily, weekly, monthly level. As of now, we don't know how people's responses would change over those different periods of time. If you said, yes, I feel really purposeful today, does that mean you'll feel that way tomorrow? So using 4 different data sets, ranging from hourly, daily, weekly, and monthly report, and over 4,000 participants, my goal is to understand how much sense of purpose fluctuates within an individual. And this will allow for 2 things. First, by understanding to what extent this construct fluctuates, we'll then have a better idea of how challenging it is to intervene upon it. For constructs that are more stable over time, those are harder to change. But if we see that at the daily level, people's sense of purpose varies quite a bit from one day to the next, then we'll then be a better position to intervene upon it. And the second thing here is connected to time. If we know how much this construct is, or if we know whether this construct is fairly stable at the hourly level, but it changes a lot from day to day, then we're in a good place to figure out what exactly is tied to these changes in sense of purpose. So is it connected to having positive social interactions or engaging in meaningful activities? So my dissertation will set important groundwork to understand how we can intervene upon it when we need to intervene upon it, so we can help older adults experience successful aging. All right, Michelle, thank you.
Thanks. Yeah, so how will you go about measuring sense of purpose? Like what kind of metrics are you gonna be looking for?
Yes, yeah. So It differs a little bit depending on the study, because an issue that we run into is how time fits into it. Obviously, that's why we're doing this whole thing. But participants will fill out surveys. So for the hourly survey, participants every hour just going to fill out an item that says how purposeful they feel. For some of the longer metrics, like the monthly survey, sorry, the slide got a little bit messed
sorry
up.
about that.
For the monthly survey, participants are actually filling out a sense of purpose measure that includes 4 items. Everything's measured on a Likert scale. So like 1 strongly disagree to 7 strongly agree. How well does this describe how you feel? And an item from that is my activities align with my true interests and values. So the questions I asked at the beginning, those are actually some items from the measures we'll be using. Becca?
I was curious how if you've seen a bias in participants based off of their participation in the question.
So like, if people are leaning more purposeful simply because like desirability, like social
Like almost
desirability
if
bias, or-
participation in
Ah,
the study is giving them a sense of purpose.
oh, interesting. Okay, yeah, so I think this could actually work. That's a really interesting question. I think this could actually work in a couple different ways. My idea would actually be that it would make people more extreme. So if you typically feel like you have a low sense of purpose and then you're answering these questions all the time and then being forced to think about, wow, nothing I'm doing really matters that much to me, then I think that could actually make it more intense on either side. That's a great question.
Okay. One more quick. Nancy, did you have one real quick? Or Emily, you have one? Okay.
Have you identified a biological pathway for how a sense of purpose affects aging, or is that the purpose of your thesis? Yes, so to this point, there hasn't really been much biologically. A lot of the work on sense of purpose is focused more on what people do. So we see that people with a higher sense of purpose engage in a lot more health behaviors. They're more likely to eat their vegetables, to floss, to exercise, to use preventative health services. However, biologically, there hasn't been much work to this point, but that's a really important question as well.
All right. Thank you very much.